All right, welcome to everyone who's just logging on. We'll give it just a moment here for people to get connected. All right, hello everyone and welcome to today's SSR webinar. If you have any questions about this webinar, please feel free to reach out to us directly via email at membership@ssr.org. This webinar is scheduled for 60 minutes and it is being recorded. It will be posted on the SSR website shortly thereafter for access at your convenience. In just a moment, we'll have a brief introduction followed by the speaker presentation. Please feel free to submit your questions during the presentation using the Q&A text box in the bottom middle of your screen, and our moderator will read them to the speaker when the question portion of the webinar begins. With that, I will turn things over to Romana.
Right. Thank you very much, and welcome to everyone who's here for this wonderful presentation we're going to have today. I'm very pleased to introduce our speaker, who is Dr. Amanda Sferrucci-Perry. She's the senior author on the paper published in BOR last year, 2022, which won the top research science article award. And what that means is that this paper, her paper, was the most highly cited paper in BOR from 2022 at the time that we made the selections. I'd like to just read you the title and also the other authors to acknowledge the other authors who, of course, made important contributions. So the title of the paper is Placental Structure, Function, and Mitochondrial Phenotype Relate to Fetal Size in Each Fetal Sex in Mice. And the first author is Dr. Salazar Petres, and also Pereira Carvalho and Lopez Teo. And I'm sorry if I mispronounced. So they're the co-authors on this paper. I'd like to just give a very brief little introduction about our speaker, because she's got an amazing bio. So Dr. Sferrucci-Perry got her degree, her PhD in Australia, and then made it across the pond over to Cambridge. And she's had a series of extremely prestigious fellowships, research fellowships, including the Biomedical C.J. Martin Research Fellowship, Next Generation Fellowship from the Center for Trophoblast Research, where she's still an active member. She also received the Dorothy Hodgkin Research Fellowship from the Royal Society, and also was a fellow Lister Institute for Preventative Medicine fellow. So a number of very prestigious fellowships, and her career has just really taken off there. She is currently the University Professor in Fetal and Placental Physiology in the Department of Physiology, Development, and Neuroscience at Cambridge University, and it's also a member of St. John's College. And so I'm very excited to have her present her work from this prize-winning paper, and I'll turn it over to our moderator, who is Haijun Gao, I believe. Yes?
Yes, welcome everyone. Thank you for your coming. So I'm Haijun Gao. I'm a SSR Virtual Education Committee member. So I will be the moderator today. So I hope you will enjoy the wonderful talk. So thank you, Dr. Novak's introduction. So now we'll give time to Dr. Amanda Sferrucci-Perry.
First of all, I'd just like to say thank you so much to Haijun and Romana and SSR. Oh, sorry. I'll just get my presentation up. Hopefully you can see that. So I just wanted to say thank you so much to Romana, Haijun, and SSR and Biology of Reproduction for the opportunity to tell you a bit about my lab's research. And on the back of our Biology of Reproduction paper, which I'll feature as part of this talk, just to say that that work was conducted maximizing samples right in the pandemic, and it involved 3 talented researchers, all from elsewhere, one from Brazil, one from Chile, one from Spain, from a variety of backgrounds, including being a vet. And so it was really a labor of love and collaboration, and that will feature in this talk. And I really look forward to answering any questions that you might have about it. So as you may know, around 140 million babies are born each year, and that's around 4 births every second of every day. And of course, the expectant parents hopes that their baby will be born a good size, a healthy size, that will have a higher chance of surviving and thriving postnatally. However, unfortunately, around 1 in 10 pregnancies will be complicated by conditions affecting the way in which the fetus grows. For example, babies may be born.
Amanda, we're not able to see your slides, it looks like, from the audience. Would you be able to just pause the screen share and restart?
Yeah, sure. Okay, so
sorry about that.
Pause, share, and then resume share. Oh, hang on. Now it's not sharing again. Sorry about that. Let me go back to my slide. Can you see my screen now?
Not just at the moment.
Okay. Screen sharing has failed to start. Please try again. Okay, let's do it again. Sorry about this. It won't let me share for some reason. It was fine earlier. Let's try again. Can you see my slide?
No, not seeing them at the moment. Would you like to email them to me and I can see if I can pop them up for you?
Yeah, I can email. It's just I like to click through.
Yeah, of course.
But I can email them to you. Let me try and screen. Why won't it? Thanks. Screen sharing has failed to start. Start again later. If it's a dual display, sometimes you have to drag it from one screen to another. Okay. Let me see if I do that. I'll put it like this. And sorry about this. It won't let me share. I can email the slides. Just gonna be a bit of a thumb first. Clicking through. Sorry about this. Adrian, I'm going to email it to you, okay?
Yeah, if you wanted to try possibly leaving and rejoining might just reset
as an alternative,
for you.
sure.
It was definitely working for you before.
Yeah, it's quite a bit amusing.
Thanks everyone for just hanging in here with us for just a second while we get this up and going.
And then I'll leave and come back in as well. It works fine earlier. I don't know why.
So Amanda, can you close your PowerPoint and then restart it?
Sure. And I'll close it and then I'll restart it. And then I'll try and share my screen. No, it won't let me share my screen. If I exit and then come back in. I'll leave the meeting and I'll come again. Sorry about this.
Yeah, of course, no worries. Thanks
Sorry,
again.
everyone. So yeah, Amanda tried before the meeting. Everything worked well. So I don't know what's wrong there. So thank you for your patience.
Okay, I'm back. Let's see if I can share my screen.
Perfect. I'm able to see that now.
Oh, amazing. Okay, let me just put it into presenter mode so that you can see the full slides. And let's just change this. Can you see my screen?
Yes, that looks great.
Oh, excellent. Sorry about that. So as I mentioned, around 140 million babies are born every year worldwide. That's around 4 births every second of every day. And of course, the expectant parents really want the baby to be born of a healthy weight with high chance of surviving and thriving postnatally. However, my lab is particularly interested in understanding what might go wrong in a pregnancy that may affect fetal growth and birth weight. This is because around 1 in 10 pregnancies will be complicated by conditions that impact on the growth of the baby during pregnancy. These can result in overgrowth of the baby, where the baby has received too many nutrients, or it can also result in fetal growth restriction, or small for gestational age, where the baby hasn't received enough nutrients and oxygen to grow properly. We know that these babies are at higher risk of poor health during poor survival prenatally, and that the changes in fetal growth can accompany alterations in body proportionality. For example, babies that are born growth restricted, they often tend to show brain sparing effects where the brain is preserved at the expense of other organs like the liver and the systemic body organs. Whereas in the case of macrosomia, or large for gestational age, the baby may have more adipose tissue. We also know that these babies have a lower chance of survival at birth, and there can also be maladjustments to life outside of the womb, where they may struggle to breathe, to grow properly. We also know that altered fetal growth and altered birth weight increase the risk of neurological and metabolic problems during childhood, as well as increase the risk of the baby to develop life-shortening conditions in later life. So what are the factors that can contribute to fetal growth outcomes? Well, work in human populations, but also in a variety of experimental animals, have shown the high importance of an adequate maternal environment in governing fetal growth. Data have shown that maternal nutrition, stress, oxygen levels, cardiometabolic diseases can all impact on the way in which the fetus receives nutrients and grows during pregnancy. And this can result in permanent changes in the structure and function of individual organs. Moreover, these organs can be programmed such that they dysfunction and have greater susceptibility to pathological changes in later life, in childhood and adult life. Furthermore, work has suggested that this programmed alteration in the offspring as a result of course, gestational environment can sometimes depend on the postnatal environment, if there is a big mismatch in the prenatal and the postnatal environment. And there can also be differences in the outcomes of the offspring depending on whether they are male or female. Another vital determinant of fetal growth, and through which the mother's environment likely mediate effects on the fetus, is this organ, the placenta. The placenta is an external fetal organ that connects the fetus to the mother. It's essential for pregnancy because it performs many vital functions, including anchoring the fetus to the mother. It recruits a blood supply, and it is intimately connected to how the fetus is growing during pregnancy. It also serves as an important barrier to the mother's immune system, preventing immunological rejection of the fetus. It also is responsible for removing waste and detoxifying any xenobiotics, providing an excellent barrier for the fetus to grow and develop properly. The placenta also secretes an array of factors, including hormones, into the mother that signal to the mother so that she can accommodate pregnancy, so that she can allocate resources, nutrients, and oxygen to the fetus for growth. And finally, the placenta is responsible for transporting all the nutrients and oxygen a fetus needs to grow and develop. And so my lab has been really interested in understanding the extent to which the maternal or the gestational environment and placental function interact to determine growth outcomes of the female and male fetuses. So today I'll tell you about three main stories. The first is our work that was published in Biology of Reproduction that Romana talked about, where we studied how the placenta may respond to natural variations in fetal growth during normal and compromised pregnancies. I'll then tell you a bit about our work looking at how the placenta may respond to suboptimal maternal environments and what that might mean for fetal growth outcomes. And I'll tell you about our work looking at pre-gestational obesity. However, we've looked at many environmental challenges, including hypoxia, advanced maternal age, just a snapshot of many factors that we're looking at. And then I'd like to tell you a bit about our most recent work, where we've been exploring how the placenta might be communicating to the mother, allowing her to support fetal growth during pregnancy, and what that might mean for later life outcomes for the offspring. So I highlighted the multifunctional nature of the placenta, which also has to grow and serve both the mother and the fetus in pregnancy. And this growth and function of the placenta is heavily dependent on energy. And that energy is primarily provided by mitochondria. Now, mitochondria are multifunctional organelles within the cell that produce energy by oxidative phosphorylation using electron transfer chain complexes embedded within the inner mitochondrial membrane. However, they also integrate several metabolic processes, including the citric acid cycle and beta oxidation of fat. They also produce reactive oxygen species as a byproduct of OXPHOS. They are also very pivotal in steroid hormone production and play a critical role in signaling and the execution of apoptosis through modulating calcium levels. Mitochondria are also very dynamic organelles because they can replicate by biogenesis, they can divide by fission, and combine by fusion in response to different cues. With this in mind, we were really interested in understanding whether changes in mitochondrial function relate to placental structural and functional support of fetal growth. Now, to do this, my lab primarily works in the mouse as a model because it has a short gestational length and lifespan, and it has a special type of placenta called a hemochorial placenta, which is similar to humans. So hemochorial meaning that there's just the placental trophoblast that separates the mother's and the fetal blood. Also, because it's a litter-bearing species, you have natural variation in fetal weight within the litter, even though the mother's environment is controlled and is not associated with any placental pathology. Furthermore, the placenta, which is shown here, is beautifully organized into functionally distinct regions. You have the labyrinthine zone, which is vascular and performs transport, whereas the endocrine zone, which cups around the transport zone, is functions in hormonal support of pregnancy. You can separate these and you can study them in a very precise and molecular way, which has been really beneficial for our research questions. So we set up matings of just normal wild type mice, and then in late gestation, on day 18 of pregnancy, which is about 90% of gestation, because pregnancy lasts about 20 days in our mouse strain, we then obtained the microdissected labyrinth transport zone from the heaviest and lightest fetuses of each sex. We then assessed the morphology of the transport region, actually in whole placentas. We also then performed mitochondrial energetic analysis using high resolution respirometry to look at the coupling of substrate use to energy production. And we also measured the abundance of a variety of mitochondrial related factors, nutrient transporters, hormone handling, and signaling pathways by real-time PCR and Western blotting. This figure just shows you that our lightest of each sex is in fact lighter than the heaviest of each sex within the litter. So you've got the heavier of the fetuses in the darker color, and then the lighter in the lighter color, and the females are in purple and the males are in green. And what you can see is that there is a weight discrepancy in the weight of the fetuses between the lightest and heaviest, which you would expect. However, interestingly, the actual proportionate growth of those fetuses was similar. We saw a similar ratio of the brain to body weight, liver to body weight, which are important indicators of symmetry of growth. We also found that there wasn't actually any difference in the size of the placenta, the size of the transport zone, or calculated efficiency of the placenta or the transport zone relative to the weight of the fetus. And this really supports the notion that these are healthy pregnancies, and we're really just looking at the disparity in fetal growth within the litter and how that may be mediated via changes at the level of the placenta. So we wanted to know whether there could be any structural changes at the level of the placenta that may relate to the lightest of each sex. You can stain the placenta and localize and assess the abundance of trophoblast, fetal capillaries, and maternal blood spaces in this transport region. Then using stereology to precisely determine their compartment volume and the surface area for exchange, as well as the barrier to diffusion, we found that overall, the composition of the placenta was affected in a sex-specific way. We found that the lightest, the placenta supporting the lightest female had a smaller volume of maternal blood space and a smaller surface area of the maternal facing compartment compared to the heaviest female. We, however, didn't see any morphological differences within the transport region between the lightest and the heaviest males. We also undertook high-resolution respirometry to assess if mitochondrial respiratory capacity could be altered between the lightest and heaviest of each sex. This involves adding in substrate, uncouplers, and inhibitors of individual complexes of the mitochondrial electron transfer system to inform on fatty acid oxidation, oxidative phosphorylation, and total capacity for electron transfer. What we found was, again, that there was a sex-specific difference in mitochondrial respiration between the lightest and the heaviest fetuses within the litter. In particular, we found that there was an upregulation, adaptive upregulation in complex 1 supported leak and coupled complex 1 and 2 supported mitochondrial respiration in those lightest, in those placentas supporting the lightest females, suggesting it may be adapting to try and optimize fetal growth. However, we didn't see any change in mitochondrial respiratory capacity between the lightest and heaviest males within the litter. Now, although we only found changes in mitochondrial respiration for the lightest females compared to their heaviest counterparts, we did find that there were changes in the abundance of mitochondrial-related proteins. For example, we found that the abundance of mitochondrial complex 1 was lower in the lightest females compared to the heaviest females, whereas in the case of the males, it was actually a reduction in complex 3 and complex 5 ATPase that was seen between the lightest and the heaviest males. Now, given that mitochondrial respiration was either increased or unchanged, this would suggest that there's enhanced mitochondrial efficiency to produce energy for the placenta, supporting the lightest compared to the heaviest fetus of each sex. We also saw sex-specific differences in the levels of genes that control biogenesis and fission. For instance, in the females, there was a suppression of mitochondrial biogenesis marker and fission markers, whereas in the case of the male, we found there were greater expression of a biogenesis marker in the placenta supporting the lightest male compared to the heaviest. How these link back to the changes in mitochondrial respiration or placental support of fetal growth require further study. But we wanted to know more about how the placenta may be supporting the fetus based on its weight within the litter. So we measured the levels of important transporters for amino acids, glucose, and lipid transporters in the lightest of each sex compared to the heaviest of each sex. And what we found was that amino acid and lipid transporters were not varied between the lightest and the heaviest. However, in the case of the males, glucose transporter expression, SLC2A1, was down for the lightest compared to the heaviest males. And this may underlie the lower weight of the fetus seen. This wasn't, however, seen for the female. We also saw differences in the level of steroid metabolic and signaling genes within the placental transport region, depending on sex. For example, we found in the lightest female, the placenta supporting the lightest females in the litter, there was greater expression of CYP11A1 and lower expression of CYP17A1 compared to the fetuses, the heaviest female fetuses. And these would favor progesterone and estrogen production and may have some explanation for changes at the level of support of the fetal development. We also found that there was elevated expression of androgen receptor by the placenta of the lightest males compared to the heaviest. This would enhance androgen sensitivity. And there are some data that suggest that overexposure to androgen might affect mitochondrial biogenesis and respiratory function. And so it'd be interesting to know whether, in fact, fetal androgen signaling to the placenta might underlie some of the changes that we saw in the case of mitochondrial capacity. We were also keen to try and understand if the changes in the placental support of fetal growth between the lightest and heaviest of each sex might relate to the activity of important signaling pathways within the transport zone. We know that growth signals like insulin operate through the AKT signaling to mediate downstream effects such as the upregulation of nutrient transporters. We also know that energy deficit and other nutrients can impinge on the activity of AMPK with widespread effects on metabolism within the cell. What we found was that an important adaptation of the placenta supporting the lightest of each sex was an increase in the expression of total AMPK. However, the difference was more pronounced for the female than the males compared to their heavier counterparts. We also found that in the lightest males compared to the heaviest, there was reduced activation of AKT, which may suggest compromised insulin signaling, perhaps from the fetus that's growth compromised to the placenta. We also assessed the expression of this important transcription factor called PPAR gamma, because it also functions as a nutrient sensor and controls fatty acid synthesis at the level of the placenta and regulates the formation of the transport region. And we found that that PPAR gamma abundance was greater in the lightest compared to the heaviest female, but was lower in the lightest compared to the heaviest male. How this may relate to placental lipid handling is unclear, but may have implications for lipid supply to the fetus and fetal growth outcome within the litter. So with this work, we could conclude that placental structure, function, and mitochondrial phenotype certainly relate to size of the fetus within each fetal sex in mice, and this operates via sex-specific changes at the level of the morphology of the transport region, signaling pathways, mitochondrial dynamics, mitochondrial energetics, and expression of genes that regulate mitochondria and nutrient transport pathways. But we've also been very interested in how the placenta may respond to support fetal growth in the context of suboptimal maternal environments. We've been particularly interested in diet-induced obesity because it's probably no shock to you that the rates of overweight and obesity, as indicated by the red, are increasing worldwide in many countries, where you can see compared to 1975, back in 2016, the red is encroaching in many, many countries. And what this means is that the proportion of women entering into pregnancy overweight or obese is also increasing. In the UK and in the US, fewer than half of pregnant women have a body mass index within the normal healthy range. And this is concerning because obesity increases the risk of pregnancy complications like gestational diabetes, preeclampsia, preterm birth, and even stillbirth. It also increases the risk of fetal growth and metabolic problems, where babies can be large or macrosomic. They can also be small for gestational age, and they can have problems with adjusting to life outside the womb. So we were quite interested to understand the mechanisms underlying fetal outcome in response to maternal obesity. And so using mice, we fed them either a control diet or a high sugar, high fat diet that contained about 3 times as much fat and about 5 times as much sugar. We fed it from 6 weeks prior to pregnancy and then during pregnancy after mating with a control diet fed male. We then assessed resultant effects on the fetus, its growth, glucose levels, as well as placental structure, mitochondrial respiratory capacity, and mitochondrial-related proteins. What we found was that in the case of diet-induced obesity, fetal growth was restricted. We saw a reduction in fetal weight for both the male and female fetuses, maybe slightly stronger for the female fetuses. And this was associated with a greater number of fetuses being classified as small for gestational age, as well as a concomitant reduction in the number of fetuses that would be classified as large for gestational age based on the normal distribution of the control population. We also found that not only were the fetuses growth restricted, but there were signs of altered body proportionality. For instance, the ratio of fetal brain to body weight was overall increased in obese mothers, and this effect seemed to be more pronounced for the male fetuses than the female. Interestingly, this was not related to a drop in fetal glucose levels. If anything, the fetuses received more glucose than in an obese animal than in a control animal. We found that fetal growth restriction was coupled to defects in the formation of the placenta. We saw a very dramatic reduction in placental weight, where you can see both the male and the female of obese animals growth restricted. However, the female placentas were overall lighter than the male placentas. We also found that there was a specific reduction in the transport labyrinthine zone of the placenta. However, this effect seemed to be more pronounced for the males compared to the females in these obese animals. We also saw that there were morphological defects in the transport region. In response to diet-induced obesity, we found that there were reductions in the volume of the trophoblast and the maternal blood space, effects that seemed to be more pronounced for the placenta supporting the male fetuses, whereas the vascular surface area of both the maternal and fetal compartments, as well as the length of the capillaries in the placenta, were all suppressed in the diet-induced obese animals. This is in line with the reduced fetal growth that we saw. We also saw that there was an interaction effect of obesity and sex to regulate the thickness of the barrier for diffusion. We found that in obese animals that the placenta of the male had a thicker barrier to diffusion, and this was associated with the reduction in the oxygen diffusion capacity of the placenta. Now, because hypoxia is a key stimulus for the fetal brain sparing effect, we suspect that this may underlie that greater fetal brain to body weight ratio we saw in the male. However, we did see some sex differences overall, where the male placentas had a greater density of fetal capillaries and had a thinner barrier to diffusion compared to females. However, this was lost in the case of obesity because of the effects on the male. We performed high-resolution respirometry on the transport region, like I had mentioned previously, looking at the different components of the mitochondrial respiratory chain. And we were very surprised to find that despite morphological defects in the transport region, mitochondrial respiratory capacity seemed unaffected by diet-induced obesity. If anything, we found that the male placentas overall had a greater capacity for fatty acid oxphos, complex 1 and complex 2 supported respiration, and overall a greater electron transfer system capacity compared to females. However, this difference seemed to be magnified in the diet-induced obese animals. Now, the maintenance of placental oxphos capacity in the obese animals seemed to link to sex-specific differences in mitochondrial-related proteins. We found that in the case of the males, there was greater expression of electron transfer system complexes 2 and 5, whereas in the case of the females, there was greater turnover of mitochondria, a greater expression of the biogenesis marker PGC1 alpha. We also were interested in knowing whether there could be changes in placental lipid handling and PPAR gamma signaling. As I mentioned, PPAR gamma is really important in regulating fatty acid synthesis. It also controls the activity of this enzyme called ACC, which is an enzyme within the mitochondria that's needed for fatty acid synthesis and inhibits the oxidation of fatty acids. So we looked at PPAR gamma and ACC activation in the transport regions of our diet-induced animals, and found that in the case of the male fetuses of obese animals, there was an increase in the inactivation of ACC, which would have led to a reduction in fatty acid synthesis and maybe a greater allocation of lipids to the fetus, and maybe a less severe reduction in fetal growth as what we saw compared to the females. Whereas in the case of the females, we didn't see any change in ACC activity, but we did see an upregulation in PPAR gamma levels, which would favor fatty acid synthesis by the placenta. Again, very different effects seen with obesity in the two sexes. We also found that maternal obesity was associated with the activation of oxidative stress pathways in the placenta. We stained for an indicator of lipid peroxidation and found that overall obesity increased the levels of oxidative stress, particularly in the transport region. And this was really prevalent in the placenta supporting the male. And it might link to the greater but however, overall, there were greater levels of oxidative stress in the males compared to the females. And this may link to the greater respiratory capacity of the mitochondria in the males compared to the females, because the males are the mitochondria are a main source of reactive oxygen species within the cell. We also found other indicators of oxidative stress that were particularly prevalent in the males. We saw greater accumulation or deposition of calcium within the transport region of the males supported by obese animals, which linked likely to the greater oxidative stress, but may have implications for the symmetry of growth and the support of fetal growth in those pregnancies. And this allowed us to conclude that diet-induced obesity certainly impacts on fetal placental growth via sex-specific alterations in placental morphology, mitochondrial respiratory capacity, lipid metabolism, oxidative stress signaling to modulate the growth outcomes of the fetus. But the placenta is also a source of signals and may influence the maternal support of fetal growth. And we have recently been looking at this very precisely in the mouse using a genetic approach. The way in which we've been doing that is to alter the junctional zone, the endocrine cells of the placenta, using a special Cre line called TBPA Cre. We're using this Cree line to induce the loss of an important growth factor called IGF-2 just in the endocrine cells of the placenta that are responsible for secreting hormones into the mother. We chose to manipulate IGF-2 in the placental endocrine cells because it's a paternally expressed imprinted gene, meaning that the mother's copy is silenced by imprinting, and it's really highly expressed by the fetus in the placenta in virtually all of the mammalian species studied to date. Also, defects in the IGF-2 locus are associated with major fetal growth syndromes, like Becker-Weiss-Vitamin syndrome and Silver-Russell syndrome in humans. It's also associated with other growth syndromes in other species like cow, as an example. Moreover, we and others have found that expression of IGF-2 by the placenta is altered in human pregnancy complications like large for gestational age, fetal growth restriction, and small for gestational age. And importantly, IGF-2 is thought to increase placenta endocrine cell formation and output in rodents. So what we have been doing is deleting the copy of IGF-2 inherited from the father in the endocrine region of the placenta, leaving the transport zone, the fetus and the mother unaltered. And using litters of mixed genotype and then doing real-time PCR on the separated zones of the placenta, what you can see is that IGF-2 is indeed reduced in the endocrine zone and not the transport zone of the mutants compared to wild type. And in situ hybridization also confirmed this to the junctional zone. But what we found so striking was that despite a lack of IGF-2 in the endocrine cells, there wasn't a change in the size of the endocrine region. There wasn't also any change in the circulating levels of IGF-2 within the mother. Instead, we found that a loss of IGF-2 from the endocrine region affected the hormone production capacity of the placenta. So we obtained cells, trophoblast cells, from our control and our IGF-2 loss placentas. We cultured them and took these supernatant and undertook proteomic analysis. We detected about 1,400 proteins in the media, of which around 290 were either absent or present in the media of the control versus the IGF-2 mutant placentas. And just focusing on those 290 proteins, when we perform pathway analyses, we found that these are implicated in reproductive, hormonal, and metabolic processes, including prolactin, soluble leptin receptor, and TNF-alpha. And these hormones are really interesting because we know that they're important for modulating insulin production and insulin sensitivity, and therefore may be a means of manipulating insulin handling by the mother in pregnancy, and thus the allocation of nutrients to the fetus. We also performed single nucleus sequencing of the dissected transport region to see how IGF-2 loss may be affecting the endocrine capacity of the placenta. And we found that overall genes were largely downregulated, and this was in a cell-specific fashion, where, for example, the spongy trophoblast and glycogen cell lineages had very profound changes, including in the expression of several members of the prolactin superfamily. We also identified that several of the genes that were dysregulated by loss of IGF-2 related to translation, ribosomal biogenesis, related to mitochondrial respiratory capacity, indicating indeed that the loss of IGF-2 is affecting the ability of the placenta to synthesize and produce hormones. And in fact, the metabolic consequence of the loss of IGF-2 was illustrated by looking at levels of critical energy sensors, pipa gamma, as well as looking at ribosomal translation-related protein in the dissected transport zone. But what I find so interesting is that what impact might this have on the maternal ability to support pregnancy? So we set up crosses of mice to compare normal mice that had normal litters and mice that had entire litters that lacked IGF-2 in the endocrine region, so altered placenta endocrine function, and compared these pregnant mice to non-pregnant females. We assessed glucose and insulin handling of these 3 lots of mice, and I'm just showing you firstly the result of our glucose tolerance test. So we starve the animal and then we inject in glucose at time 0, and then we look at glucose as it's cleared from the circulation across 2 hours. And I'm showing you here, compared to non-pregnant mice, pregnant mice with normal litters more slowly clear glucose from their circulation. Moreover, when we perform an insulin tolerance test, which instead we inject in insulin at time 0 and then look at the removal of glucose from the circulation, what you see is compared to non-pregnant mice, that pregnant mice with normal litters are less able to take up glucose from their circulation, indicating the acquisition of glucose intolerance and insulin resistance in pregnant mice with normal litters. However, if we look at the mice that have the mutant placentas that are not functioning properly, these adaptations fail to occur. These mice have a similar glucose tolerance and insulin sensitivity as non-pregnant mice. And this would be expected to have consequences for the availability of nutrients in the maternal circulation for transfer to the fetus. And in fact, when we look at the levels of lipids in the maternal circulation, what you can see is that these mice that lack IGF-2 in the endocrine region have placenta endocrine malfunction, that they do not increase triglycerides and cholesterol as they should as part of normal pregnancy. And they also have a lower level of fatty acids in their circulation compared to pregnant mice with normal litters. Moreover, there are consequences for the allocation, the transfer of glucose from mother to the fetus. We performed in vivo nutrient transport assays and found that the transfer of maternally supplied glucose to the fetus by the placenta was reduced in pregnancies that had the mutant placentas. And this was seen similarly for the female and male fetuses. And this was coupled to a reduction in fetal growth of about 8 to 10%. However, we're also interested in the outcomes of the placenta endocrine malfunction for offspring health. And so we let the litters deliver naturally, and then we looked at the growth and metabolism of the offspring into adulthood. The offspring were born smaller, but they rapidly caught up. But what we found was so striking is that at 12 weeks of age on a chow diet, the female and male offspring, remember these are normal, they were just supported by a mutant placenta, they show an inability to fully lower glucose in their circulation compared to offspring that was supported by controlled placentas in pregnancy, indicating that offspring are programmed for insulin resistance as a result of placenta endocrine malfunction. We also went on to show that the offspring were more insulin resistant and had a greater propensity to gain excess adiposity on a high sugar, high fat diet from weaning, indicating it amplifies this programmed effect of a problem caused by the placenta. So with this work, we could conclude that the placenta indeed signals to the mother through the production of hormones to adapt her glucose and insulin handling, so she frees up lipids and glucose for transfer to the fetus for growth. Moreover, if this is not adequately undertaken, this can result in programmed outcomes for the offspring where they have an elevated risk for obesity and insulin resistance. So I hope that I've convinced you that placental structure and function alter developmentally to the needs of the fetus for growth during normal pregnancy and in response to suboptimal gestational environments. Moreover, the mechanisms mediating developmentally and environmentally induced responses of the placenta include metabolic genes, signaling pathways, and mitochondria. And importantly, these are influenced by the sex of the fetus, which cannot be ignored. Furthermore, the placenta also secretes factors into the mother that promote fetal nutrient allocation and growth, with important implications for postnatal offspring health. And we hope that this work could inform the design of therapies targeted, for example, to the placenta and tailored to the sex of the fetus for complicated human pregnancies. And we do hope that they overall advance our understanding of the pathways underlying poor fetal growth outcomes and their consequences for health and disease risk. And although I mentioned our mouse work, we also try and translate our findings and apply our strategies for the mouse on human pregnancy samples. And so I've got our papers here in case if you're interested in some of our human-related work. And it just leaves me to acknowledge my great lab, particularly those circled here, who have contributed extensively to the work that I've shown you and for which it wouldn't be possible. My main collaborators in Cambridge, my funding bodies, and just to say that in Cambridge, we have several initiatives that have been designed to support people's interest in the placenta and reproduction. You're welcome to get in touch with me if you've got any interest in these courses, and I'd happily tell you more about them. And just to say, I'm happy to take any questions and happy to follow up via email, and certainly always happy to collaborate with those who have an interest in work and sharing knowledge and expertise. And thank you for your attention. I'll stop sharing now.
Thank you very much, Amanda, for this wonderful talk. So I bet your work will provide a foundation for future studies in the placental structure, function, placental programming, and many other research topics in the field of reproduction. So really appreciate this wonderful work. So I received some questions. So the first one, some audience tried to request these PowerPoint slides from you. So I might give them your email and let them request you directly later, okay?
Yeah. No problem.
Okay, so there are some questions about the animal model you used, especially for the obese mice model. So the first one, how long you treat the mice with the high-fat, high sucrose diet before they get obese?
Yeah, so that's a really important question. With the story that I showed you today, the mice had been fed the high sugar, high fat diet from 6 weeks before pregnancy and then during pregnancy. We know that the mice enter into pregnancy obese. They have about 3 times as much the adipose tissue. However, we have done other studies, and I'm happy to share those papers with you, where we have fed the mice the diet much longer for about 9 to 12 weeks prior to pregnancy. And then we've also done shorter term exposures of just feeding the diet from the start of pregnancy. I think that the length of time that the diet is fed for and the composition of the diet is really important. In our hands, we always get fetal growth restriction. We don't ever see overgrowth, which is often seen in women who are obese. But I think that the composition of the diet is really important to consider in terms of what fetal outcomes you might be studying.
Okay, another related question is, are the obese mice diabetic before they get pregnant?
So that's a very good question. Actually, we haven't checked in this cohort of animals whether they enter into pregnancy with altered glucose or insulin handling. We do know that they are glucose intolerant though, and they have higher levels of insulin in pregnancy, but we did not look prior to pregnancy at what, aside from adiposity, but that is something really important to know to understand what the metabolic physiology of the animal at the point of entering into pregnancy.
Another question is asking you about, did you perfuse the animals before tissue harvesting? If so, does it affect placental morphology?
So that's a very good question. We don't perfuse our animals. We normally just drop fix in samples, mainly because we use our animals for multiple purposes. So if we were to perfuse them, say, for example, with fixative, we would only be able to use the placentas for histology. And within any litter, we try and get information also about the not just histology, but molecular changes. But certainly if you are doing perfusion fixation, and we have done that on select animals, but not on the ones that I've presented here, knowing the distance to perfuse in and making sure that that is physiological and doesn't disrupt the delicate tissue architecture is going to be important.
Okay.
But we haven't directly compared perfused versus non-perfused animals in terms of structure, which I think would need to be really carefully looked at if people are going to go through the notion of doing perfusion fixation. Okay,
one related question is about the tissue collection. So you use the frozen tissue to measure the mitochondrial function, right? So respiration specifically. So did you develop this protocol? Or another question is, did you use this for other tissues?
So that's a very good question. So we looked at, so we did freeze the placenta, but it was cryopreserved. So we first drop the placenta and expose it to a cryopreservation media, which preserves the mitochondria and keeps them alive. And it also helps to freeze the tissue very slowly. We then thaw it very slowly and do a series of washes before studying the mitochondria, which are intact and within the tissue, which is really important because we're studying mitochondrial respiration within the tissue, not of isolated mitochondria. The placenta is quite unique that you can cryopreserve tissue for mitochondrial studies. It's been done on mouse and human placenta. And I believe there's some data coming out on rat placenta. I don't know if there are any data of other organs showing that this cryopreservation method works. A way of getting around working with samples that are frozen is to instead isolate the mitochondria and then freeze the isolated mitochondria. However, isolated mitochondria can behave differently than if they are in a tissue where they're connected to other components like the endoplasmic reticulum and normal substrates within the cytoplasm, where it's more physiological.
Wonderful. I think your novel technique will really provide the convenience for this kind of studies, especially for the samples from the patients. So usually we cannot collect enough human samples at all time, right? So I think this method is wonderful. Thank you very much.
I'm happy to send if people email me, I'm happy to share protocols for sure.
Okay. So I think we have a last question. So it's about the ratio of the male and the female in the litter. So whether the distance between the female and the male will affect the phenotype, especially for the females.
Sorry, say that again.
So like the distance between the female to the male within a liter. So
yes.
this is might affect the phenotype in the female. So
It's true. So we haven't assessed intralitter signaling, and unfortunately, whenever we do our analyses, we haven't been systematic in identifying the position of a fetus to another. But for sure, you can get some degree of signaling of siblings to one another. We have found, for example, in the case of genetic manipulations, that having a mutant within the litter affects the wild type within the litter, regardless of how physically close they are within the fawn. But that is an important consideration. I think in order to really address that, you need lots and lots and lots of animals, because you get variability from litter to litter, and in terms of within the implantation sites and where the male and females are positioned. But it is an important question to address that we haven't done ourselves.
Okay, I think yeah, there's some audience are interested in the opportunities at Cambridge. So I think you'd better contact Amanda directly. So wonderful. I think we wrap up today's seminar. So look forward to your wonderful future research, and also the opportunities to give us another talk in near future. Thank you all.
Sure, no problem. And yeah, please, I see that there's lots of questions in the chat. If people would like, I'll try. If people would like to email me, I can email them in response to their questions, because I have their names, but I don't have their email addresses.
Yeah.
But you're welcome to email me, and I will do my best to respond as quickly as I can. I put my email address again in the chat.
Okay. So I think thank you all very much. Then I look forward to seeing you in next SSR webinar.
Yep.
Have a nice day.
Thank you very much for the opportunity.
All right, great. Thank you everyone so much for your time. And attendees that are still on, we will send out a follow-up email to you all that includes Amanda's contact info if you missed it. So thank you all once again, and that will conclude our webinar for today.